All right, welcome to chapter 14 on the principles of disease and epidemiology. So before we start, a few definitions. So what is epidemiology? What are we even talking about? So epidemiology is the study of the incidence, distribution of different diseases, and the possible control of those diseases. Pathology is the study of disease itself. So this is often going to include studying different signs and symptoms of diseases, like what the duration of a disease is, what systems of the body are affected by that disease, those kinds of things. A pathogen is, of course, a disease-causing infectious agent, so bacteria, virus, protozoa that causes disease. These are all examples of pathogens. Each pathogen is going to have special properties that's going to make it be pathogenic or make it have the ability to invade the human body, possibly even producing toxins. Etiology is the study of the cause of disease. So you might think this is the pathogen, and in quite most or a lot of cases, it is the pathogen. But think like heart disease or stroke. There's lots of different diseases that don't necessarily have a pathogen that causes it. For example, heart disease is caused by obesity, genetics, a sedentary lifestyle, etc. Right? So that's etiology. Infection is the invasion and growth of pathogens in the body, which may not always cause disease. And disease is where we actually have an abnormal state in the body, where it's not functioning normally. This is when we start to show signs and symptoms, but you can still have an infection, but not be producing any signs or symptoms, so you would not be presenting a disease. Typically, once you do start to show symptoms or you develop disease, that's when the infectious agent has overcome the body's natural defenses. It also is quite often related to the numbers of infectious agents within the body. So we're going to start by looking at a few different organizations that look at epidemiology. So the World Health Organization, or WHO, is a division of the United Nations, and they promote the development of various programs, various healthcare programs, including the production and distribution of immunizations. They also help to monitor and study non-communicable diseases as well. So things like diabetes, COPD, cardiovascular disease. So it's not just diseases that are caused by a pathogen, but all diseases. But they also definitely have a large role in the response to various outbreaks caused by infectious diseases. So they will coordinate and execute any emergency response measures. They also identify and monitor threats to the global health. So for example, they have like a top 10 list of the 10 most likely diseases to be large problems in the world in the next year. They also will develop advisory guidelines. So these are guidelines for the countries to follow in response to these threats. The CDC, or the Center for Disease Control and Prevention, is more specific to the United States. They collect and analyze epidemiological information, and this is collected through case reporting. So healthcare workers are required to report if they have a patient that presents with these very specific diseases, MRSA, Zika, West Nile virus, STD, Salmonella, anything that is a communicable disease needs to be reported to the CDC so that it can be traced. The CDC also uses this data to develop morbidity rates and mortality rates for various diseases. So the morbidity rate is the number of people that are affected in relation to the total population in a given period of time. So this is the number of people that are affected. We use the word affect instead of infected here because we are referring as well to heart disease, diabetes, various diseases that aren't necessarily caused by a pathogenic species. A mortality rate is the number of deaths from a disease in relation to the total population in a given period of time. So that's the death rate, basically. Lastly, we're talking about the NIH, National Institutes of Health. This is the #1 source of funding for all research that's medical-based. So a plethora of research is funded through the NIH. This research can include development of new therapies, preventative approaches, vaccines, diagnostic tests, even just understanding the functions of the human body. Another great thing about everything that's funded by the NIH is required to be published for free. So we all have access to all of the information that has been received due to funding from the NIH. All right, we're going to switch gears here and start talking about emerging infectious diseases. So these are diseases that are new or increasing, or also have the potential to increase, or a high probability of increasing. This is caused by mutations to disease. So these various species can mutate, adapt, and change to become more pathogenic. We also see increasing disease due to increased travel and commerce. So that's going to cause any diseases that do come up to be spread much more quickly. Also due to changes in technology and industry. And what you'll know is a lot of these can all be tied back to increasing human population. So it has a huge effect on emerging infectious diseases. So changes in demographics and behavior. As our population increases, we start to live in closer and closer quarters, which of course is going to then spread disease much faster. Environmental change and land use. So we see this as our population increases, our cities start to grow in size. As we start to grow, that means we're pushing the cities past their prior boundaries, closer to like wildlife, or where different animals might be carrying different diseases. So now we're exposed to that. Also breakdown of public health measures. So as we increase our population, but we're not increasing our hospital size, we're not increasing our healthcare provider numbers. So we're overcoming these the healthcare measures that we have. So we already talked about genetics and how pathogenic diseases can undergo genetic recombination to make them more pathogenic. Also, they can undergo a natural selection, especially due to the use of antibiotics, right? Making these antibiotic resistant diseases. So we're evolving new strains of these different species. That's caused by inappropriate use of antibiotics and pesticides. So both of those are going to lead to natural selection. That's going to select for the fitter organisms that are able to survive and resist these antibiotics or pesticides. Changes in weather patterns. So this is like climate change. Zika is a great example of this. As global warming continues, we start to see it getting warmer and warmer further north. And that's why we can see mosquitoes that are living further and further north. And that's why Zika has moved up further north as well. Transportation also. So transportation can spread these different diseases. Ecological disaster, war, political upheaval, expanding human settlements, as I just mentioned. We're increasing the size of these cities. Different animal control measures, because there's a lot of diseases that are spread from animals. We're going to talk about that a little bit later. Public health failure. And then the last one is improved case reporting and monitoring. So this is a little bit different here, but it's saying that as we improve the reporting systems that we have, of course, the numbers are going to go up. And that might not be a true representation of an increase in disease prevalence, but it will be shown in the data. All right, talking about resident microbiota again. So in the body, there's microbial antagonism. This is competition between microbes. It's competition between good and bad microbes, or microbes that are good for us or pathogenic. Resident microbes, as we've learned, are microbes that are normal to our body, and they don't cause disease. The resident microbes are first acquired at passage through the birth canal, and they're further developed and modified through the foods that we eat, the people that we come into contact with, pets that we have, and various environmental factors. So your resident microbes, the different species that live on your body, are not always going to stay the same. So they're going to change throughout your life based on what you are coming into contact with. So these different resident microbes protect us in several ways. So one, they're going to occupy niches that pathogens might occupy. So this is that microbial antagonism. If you have a healthy microbiota, then it's going to have less resources and less room for a pathogenic microbe to come and invade and cause disease. So that's one of the biggest protections that the microbiota provides. Resident microbiota also produce acids, bacteriocins, and other inhibitory substances. A bacteriocin is a protein that's produced by bacteria that helps to inhibit other closely related strains of that bacteria. So it's going to help to inhibit pathogenic diseases or pathogenic species. And lastly, resident microbes also help to stimulate and train the immune system. So this is really important to help to prevent like autoimmune diseases. So this is just showing the different species that we find in different parts of our body. So we're not going to have the same microbes on all the parts of our body. So you see that we have totally different microbes on our skin than what we find in the urethra or in the large intestine. So they're all different. And actually, if we find resident microbes that should be on the skin, for example, but say that they move into the vaginal tract, they can cause disease. Or anything where we have a microbe that's healthy in one part of the body, it can cause disease if it's found in the wrong part of the body. So just take that in mind. Transient microbiota. So these are microbes that are present for just a short period of time, and then they go away. So they're not necessarily there for your entire lifetime. This is due to the dynamic nature of resident microbes. So like I mentioned earlier, your microbiota is not consistent. It's not like you're born with these microbiota and that's what you get. No, that's going to change based on the environment and your age, and what your foods you eat. Your hormones can have an effect on your microbiota. Of course, antibiotics is going to have a huge impact on your microbiota. And then probiotics is another one that's going to affect this. So you probably all heard about probiotics, which are live microbes that can be applied to. So there's probiotics for the skin, probiotics that you can ingest into the body that are to exert beneficial effects. So you're taking microbes or applying microbes that are beneficial to us to help change your microbiota. Symbiosis is any relationship between microbiota and a host. Symbiosis is any relationship between organisms, really. But in applying it to microbiology, there's three different types. So we have commensalism, which is where one organism benefits and the other is unaffected. So Staphylococcus Epidermidis is an example of this. This lives on our skin. We almost always have it on our skin, but it doesn't really affect us. It doesn't cause disease or doesn't really do anything good for us either. It's just neutral for us. But of course, it's good for the bacteria because it has somewhere to live. Mutualism is where both organisms benefit. An example of this is E. coli that lives in the gut. So this is providing benefits to us. It's helping to break down nutrients. It's protecting us against pathogens. And then, of course, we are providing nutrients for it as well as a good environment. And parasitism is where one organism benefits at the expense of another. So the example we have here is a flu virus. Of course, that's not good for the cell, but it is beneficial to the virus. Now, opportunistic pathogens are part of the microbiota that then cause disease. So remember, microbiota or microbes that are part of the resident microbiota do not cause disease, but they can cause disease and become an opportunistic pathogen for a couple of reasons. One, if other resident microbes have been destroyed, then it can cause disease. So for example, there is a bacteria C. difficile. That's not a very strong pathogen. So if we have a strong microbiota, C. difficile can live in your gut for years, and that's not going to cause any disease. It's going to live there in very small numbers. But then, if for whatever reason, your microbiota becomes unhealthy, that can happen because of an infection. It can happen because of taking an antibiotic. And now you don't have the protection from that C. difficile. And now it's going to become an opportunistic pathogen and cause disease. Opportunistic pathogens can also arise due to a weakening of the immune system. So someone who's normally able to keep that opportunistic pathogen at bay, now their immune system is weak and no longer able to provide protection, then that pathogen can become pathogenic. And it can also become pathogenic if it moves to the wrong area of the body. So an opportunistic pathogen can not cause disease in one area of the body, but then if it moves to a different region where it does cause disease, and that would be an opportunistic pathogen. Another thing is cooperation among microorganisms. So some microbes are not strong enough to cause disease on their own, but if they are mixed with another microbe, then both of them are able to produce severe enough symptoms to cause disease. All right, looking at Koch postulates. So we kind of talked about this guy in chapter 1, and I said we were going to come back to his postulates. What he did, he was trying to prove how you can determine the cause of an infectious disease. And he developed these 4 postulates or truths that had to be shown in order to prove the cause of the infectious disease. So the first one is that the same pathogen must be present in every case of the disease. So if you have several patients that are presenting this disease, you should be able to take cultures from them and identify the same exact pathogen in all of those patients. Second is that and then this is a procedure here. So the pathogen that is isolated from that host should then be able to be grown in pure culture. Then if you take that pathogen or the culture and inoculate it into a healthy, now we're going to be talking about a lab animal. You inoculate it into a lab animal, it should cause the exact same disease that you saw in the initial host. And then again, you should be able to take a culture from that secondly inoculated host, culture that, and find the exact same disease again. I think it makes more sense with the picture. So what he did, he studied anthrax. He took a mouse that had died from anthrax, and he cultured it, and he was able to identify that species. Then he took the culture, inoculated it into a healthy mouse, and the mouse presented with the exact same symptoms and the same disease. Then he cultured the disease, or he cultured the pathogen from that mouse and was able to identify that it was the exact same species as was cultured from the original sick mouse. So this exact same procedure is still used today for emerging infectious diseases. So that's all great, except for there's some modifications that have to be done. It doesn't work for every disease. So first, we can't use this for viruses because we can't culture a pure culture for viruses, right? Viruses can't live on their own. Viruses have to have a host cell. So it can't be used for viruses, and then other bacterial species that can't be grown on artificial medium. It also cannot be used for some diseases because that disease might be caused by multiple microbes. So for example, the disease pneumonia or meningitis or nephritis, they can be caused by multiple different species. It's the same symptoms, same disease, but it can be caused from different pathogens. So that's not always going to ring true for this Koch postulates. Some pathogens cause different diseases. So they might cause different symptoms in different patients. This can be due to a difference in mode of entry, can cause different diseases, or just different patients present disease differently. So that's not going to work here because then if you re-inoculated it, you might see different symptoms. And because it's unethical to re-inoculate it into a healthy individual. So it can only be used for pathogens that cause disease in lab animals. All right, now we're going to kind of go through several definitions. So classifying these infectious diseases. So a disease can be communicable. So that means that it can be spread from one host to another. Examples include genital herpes, tuberculosis, or Zika. These all can be spread. Now, contagious diseases are also spread, but a contagious disease is more easily spread than a communicable disease. So they both kind of mean the same thing, but contagious is like more easily spread, including the common cold or chickenpox. A non-communicable disease is a disease that cannot be transmitted from one person to another. So tetanus or ulcers. I was talking about heart disease earlier. It doesn't matter, you can be in close contact with a patient that has these diseases. You're not going to get that disease from them. It has other causes. The occurrence of disease. So the incident is the rate of occurrence of new cases during a period of time. So an incidence is just the new cases that are arising in a set period of time, whereas prevalence is the proportion of the population that has that disease during a set period of time. So the difference here is that prevalence is not just new cases, but it can be just people that have this disease. So think about diabetes. Incidence would be all the patients that were newly diagnosed with diabetes during a period of time. So you could say in 2020, this number of people were diagnosed with diabetes. But prevalence would be anybody in 2020 that had diabetes, whether they were diagnosed 20 years ago or diagnosed in 2020. Sporadic disease is a disease that occurs occasionally. So it pops up, can cause an epidemic, but then it's usually controlled and goes away, like Ebola or cholera. Whereas an endemic disease is a disease that's constantly present. We don't see it rise and fall. We never see it go away, like malaria or HIV. These diseases are always present with us. An epidemic disease is something that is acquired by a lot of hosts in a short period of time in a small area. And if that epidemic spreads worldwide, it becomes a pandemic. So a pandemic is a worldwide epidemic. Recent ones include SARS, MERS, or COVID-19 that we're in right now. The severity or duration of a disease develops rapidly. So like when you get the flu, you're suddenly very sick for usually a short period of time. Whereas a chronic disease is a disease that develops slower and it usually lasts longer. So like arthritis. It takes many years to develop arthritis. A subacute is a disease that's somewhere in between acute and chronic. It's not quite as acute, not quite as quick as acute, but not quite chronic either. A latent disease is a disease that has a period of no symptoms when it's inactive. So tuberculosis is an example, also shingles is an example. So shingles is caused by the same virus that causes chickenpox. So a lot of people will have chickenpox first, and then they'll go many, many years where they have no symptoms. And then suddenly they'll have shingles, or like a cold sore or herpes. These are latent diseases where you can go for a long time without showing any symptoms, and then the disease will then reappear. Also, we're going to talk about herd immunity here. So this is the presence of immunity in most of the population, and that can also provide protection to someone who's non-immunized. So this can be acquired either naturally through infection or artificially through vaccination. But where herd immunity is really important is it's the best way to provide protection for those that cannot protect themselves. So we talk about herd immunity a lot in vaccination and how important it is that the majority of the population is vaccinated, because it's the only way to provide protection for those that cannot be vaccinated. There's lots of people who cannot be vaccinated for various reasons, whether it's because they have allergies to vaccinations, they could have autoimmune diseases or other immune disorders, young babies and children before they have been vaccinated. So the only way to protect those babies is by providing herd immunity, but herd immunity only works if the majority of population is immunized or have natural immunity. All right, the extent of host involvement. So an infection can either be local, so it's limited to a small area of the body. So if like you have a cut and that gets infected, if you have like an urinary tract infection, that's going to be limited just to the urinary tract area. Whereas a systemic infection is an infection throughout the entire body because it is being spread through the blood or lymph. So once an infection reaches the blood or lymph, then that can be spread throughout the entire body. A focal infection starts out as a local infection and it spreads to a very specific area of the body. So for example, you have a urinary tract infection that can go up into the kidneys and cause nephritis. So this is a very specific spread from one local area, again, to another specific area. It's still not systemic, though. A primary infection is an acute infection, but that can cause and lead to secondary infections. So these are infections that happen due to the weakening of the host's immunity from the primary infection. So we see this a lot with COVID-19 that we're experiencing right now. COVID-19 is the primary infection, but this can lead to or cause secondary infections. So we see pneumonia a lot in our COVID-19 patients. This is a secondary infection because the lungs and the immune system has been weakened due to COVID-19. It causes the patient to become more susceptible to pneumonia. Subclinical is when you have an infection, but you don't show any signs or symptoms. We've been hearing a lot about asymptomatic carriers. This is an example of that. Toxemia is having toxins in the blood, so toxins that are produced by pathogens. Viremia is viruses in the blood. Sepsis is what happens when we have a toxic inflammatory condition. And that's going to arise from these microbes or the toxins that's in the blood. Bacteremia is the presence of bacteria in the blood. Septicemia is the multiplication of bacteria in the blood. So septicemia is where the bacteria, they are still growing and multiplying. So here's another example of a secondary infection. So you have a primary infection, whatever it may be. So you get diagnosed with an antibiotic. You start taking an antibiotic. As we mentioned, that's going to kill a lot of your resident microbes. That causes you to become more susceptible to other secondary infections. So we have a lot of yeast that live on us, which is a fungus, but our immune system is able to keep it in check with the help of resident microbes. But with those resident microbes being killed by the antibiotic, that causes the vaginal tract to become more susceptible to yeast infections. So predisposing factors. So not everyone is susceptible equally to all diseases. We see this all the time. Why does patient A get this disease, but patient B does not? Well, we have varying susceptibilities because of genetics. So our genetics can predispose us to be more susceptible to different diseases. Gender can, like for example, an anatomical male is not going to develop a vaginal infection, right? The climate and weather can have an effect on our susceptibility. So viruses in particular are spread and they grow more easily in a drier environment. Our age has a huge factor on our immune system, stress and fatigue. So if you're not sleeping well, you're just going to weaken your immune system. Different lifestyles, so eating your fruits and vegetables, getting a lot of nutrients, not just calories, but nutrients and vitamins and minerals. Exercise builds your immunity. And then other drugs such as chemotherapy or other medications can weaken the immune system as well. These are all going to cause us to change or vary our susceptibility to different diseases. All right, so all diseases have these 5 different stages. So the first stage is the incubation period. So this is the time from when you're first exposed to when you first develop the very first signs and symptoms. And that incubation period is going to range depending on each disease. Some diseases are just a matter of hours. So a lot of food poisoning can be very quick. whereas others can be a long time. That's one thing that we keep hearing about COVID-19, right? It has a very, very long incubation period, but you're still infectious during this time. Next is prodromal period. This is the first sign of symptoms, usually can be quite mild. When we have like a cold, this is that day where you're going, I don't think I'm sick, but you know, you really kind of are, because it's just kind of barely sick, right? Then you have the period of illness. This is where your symptoms are at their full force, the height of the disease. So this is where we're mostly when we say we're sick. It's the period of illness. Then we have the period of decline. So this is where our signs and symptoms are subsiding, starting to get better. And lastly, we have the period of convalescence. This is where the body returns back to its healthy state. So here's measles for an example. So here we have the incubation period of 10 days. Then we have the prodromal phase, 3 days. Period of illness is just a few days. A period of decline, you start to get better for a couple days. And then lastly, period of convalescence. So she's all better. So the different diseases are spread in different ways. They also have different reservoirs. So reservoir is the source of an infectious agent. Reservoir can either be non-living, human, or animal. Types of non-living reservoirs include soil or water. So we know that we have lots of different diseases that can be found in water, contaminated waters, as well as soil, like tetanus is found in the soil. So this is where we can find these infectious agents that can then cause disease. Humans are also a reservoir. So like the flu or COVID-19, the reservoir would be humans. So that would be either people that have that disease, or they can also be asymptomatic carriers. It's going to be somebody that is infected. So they're going to have that disease inside them. And the last reservoir is animals. So some diseases are carried by animals, like a salmonella is often found in animals. And then you come in contact with that animal and it spreads. Zoonosis, or a zoonotic disease, is a disease that can be transmitted from animals to humans. All right, so the different ways that diseases can be transmitted. So one way is through contact. And there's several different ways that diseases can be spread through contact. So all of these are contact transmission, okay? It can be direct contact. Indirect contact would be through fomites. A fomite is any non-living object that can carry different diseases on it. So anything, a table, a pen, a door handle, these are all fomites. It's somebody that had that disease touched, and then somebody else came and touched it, and now they're infected. Droplets is another contact transmission. So something is identified as being a droplet transmission if it has larger droplets. So they're not going to go as far. So they're usually just going to go just a few feet, and they're not going to stay in the air for a long period of time. This is what COVID-19 is. COVID-19 is a droplet disease. So it usually requires that you are within 6 feet of somebody, or it can also spread through fomites. And that's because it lives in larger droplets, saliva or mucus. Or congenital transmission is the last contact transmission. So this is transmission from mother to fetus or newborn. Then we also have a vehicle transmission. So it's going to use these different vehicles to transmit. So that's going to include water, food, or air. Water, you can drink it, food, you eat it, air, you breathe it. So an airborne, a lot of people get these mixed up, your droplets versus your airbornes. Something is classified as airborne if it can live in water droplets that are much smaller, or especially if it can just live in dust. And so what happens, something that's airborne can live just in the floating air for hours, can just drift around in the dust. And we don't really see that with COVID-19. It's usually going to be expelled and then settle. So that's why COVID-19 is classified as a droplet. Then we also have vectors. So we already talked about contact transmission, vehicle transmission. The last one is vector transmission. So these are diseases that are spread through arthropods, which include insects, fleas, ticks, and mosquitoes. And they're going to carry the pathogen from one host to another. There's two different ways that vectors can transmit disease, either through mechanical or biological transmission. Mechanical is just like the fly walks in poop that has salmonella, then he walks on your food and he spreads that to your food and then you eat it. So it's not actually biting you, it's just mechanically spreading it, typically through its feet. Whereas biological transmission is when it bites you. So we are going to see this quite often in ticks and mosquitoes. So it's biting an infected animal or another infected person, comes along and bites you, and then it spreads the disease to you. And then we have nosocomial infections. So these are acquired in the hospital. So you came into the hospital, you were not infected. So you came into the hospital for whatever reason, and then you get infected while you're in the hospital. Somewhere from 5 to 15% of all hospital patients acquire nosocomial infections, which is a huge amount, and it's a big problem. Using aseptic techniques can be used to prevent nosocomial infections, but that's not always the only cause. So they can be caused by the fact that there are a lot of diseases found in the hospital. So that's one. But another big one is that the patients have compromised immune systems. So we try our best to use aseptic techniques, but we have to be very, very careful because often our patients have very weak immune systems while they're in the hospital. And the last reason is there's a lot of contact with our patients. If you think about our daily life, we don't really come into contact with a lot of people, but in the hospital, one nurse is going to come into contact with a lot of different patients. So there's a lot of sources of transmission there. And it's not just healthcare providers, but it's also different tools that we use, like blood pressure cuffs, and et cetera. So just keep all these things in mind. So as we're going through this lab and you're learning about aseptic technique, I don't want you to just learn that in our lab and leave it there. The idea is that you learn about aseptic technique and then you apply that thinking throughout your life as a healthcare provider. You continue to think about everything that you touch is going to have these microbes on it. And what can you do to prevent that transmission? So here's just various diseases that are caused by nosocomial infections. So a lot of them are urinary tract infections. That's not going to be caused by the insertion of catheter. Surgical site infections, lower respiratory infections. So we see a lot of pneumonia in patients that can just be coming in for knee surgery, but they have a weakened immune system and they are sedentary as they are trying to heal. So that causes the different pathogens to settle in the lungs because they're not using their lungs as much. These are different species that cause nosocomial infections. So let's take a look at those. And also note that a lot of these are resistant to antibiotics. This is because we do see a lot of use of antibiotics as a preventative rather than a treatment in surgical patients, which we see a lot in the hospital. So we have these patients that are already on antibiotics, not because they have an infection, but just as a preventative. So they're not going to be susceptible to any ordinary diseases, but they are going to be very susceptible to antibiotic resistant diseases because now their microbiota has been destroyed and they have a weakened immune system, so they're extremely susceptible. All right, so that's the end of this chapter, and I'll see you guys soon for chapter 15.